Hello, everyone. Thank you for having me, and welcome to this third presentation of the Chemical Genomics Core facility provided by the webinar of the CTSI. Before getting into today's talk, which is RNA and DNA phage display systems and their applications in the CGCF, which is the core facility, I will break it into three different sections. And for each section, I will present and maybe make a small break. So if you have questions, or we can all discuss at the end. Before getting into the substance, let me give you the location. We are in the medical science building, the Vinyl medical science building, with those 3 rooms, one serving for higher throughput chemical and then the phage display. The core is under my direction, and some of the achievements of the core are here. And again, like mentioned, this will be published later under the CTSI website. So the core service 4 different areas. Dr. Main is in charge of the high throughput technology. Dr. Zhang is in charge of the chemical medicine. Dr. Rai is in charge of the 600 megahertz NMR. And I will be talking about the phage display, which is under my direction. But before anything, I know some people may have heard this for the first time. I really want to go as detailed as possible so you have an idea of what phage display. Let us start with the definition. Phage is just that small viruses infecting only bacteria, but we use it for technology. Phage display technology consists of an insertion of any foreign fragment, any out of the phage fragment gene into the minor structural gene of the phage, so that when the phage is expressed, this gene is also expressed and can be fused with the minor structural protein and then exposed on the phage. Once it's exposed on the phage, it's accessible, it's available for any target. And I can point to things here. You see, like, different from a recombinant protein, you have the phage genome containing the gene of the foreign protein, and then you have the platform surface exposing that protein. That's why we call it phage display. Phage that maintain entirely the capacity and then put on the surface those proteins. So you can have one example just down here. This is a normal phage, and this is a phage after they present those foreign proteins. And we have a review from 2004 and 2021 with this work. So we are working with 2 types of phages. One is the DNA phage, the M13, developed by Josh Smith. And the one we've developed lately is the Qβ. It's an RNA phage. But both will have some common methodology. It's possible to have a library insert into the minor structural protein. It's possible to just have a probe on the surface of both phages. It's possible to have the probe binding the target, or to use a library to screen any target. So this is just like roughly, and again, it's phages, it's viruses. That means they're very small. You talk about 100 nanometer on the length for M13, and then 25 nanometer for the diameter for Qβ. One is filamentous phages, and the other is just icosahedral. So those are the 2 major phages we are using, we service with. So exactly, what can we do? If you have a project that is dealing with extracellular protein interaction, we know from time to time the small protein or peptide that we use to try to map those binding domain of a concave surface of protein is very small. So that's why we recommend to use phage display. The same way, if your project is dealing with intracellular protein interaction domain, I think we can use the phage display to map those domains to the key amino acid. I'm just going to use one example that lately we've been in the pandemic, and we know one of the key protein the COVID virus is using to get into the cell is the spike protein. So the contact between the receptor and the spike protein is done by a few amino acids that we have mapped, and those amino acids can be like druggable. So this is one key example. Cell or cell-to-cell interaction through protein or microbe interacting with the cell, we can map the major domain of those proteins. One other thing, we know enzyme will have some cleft, their active portion or the allosteric portion. We can map peptide binding those portions with the technology we have. At the same time, I will talk about biosensor, maybe with the time. We can also try to target some of the key protein. We can come with a way to sense the presence of a protein or a complex through the phage display, just using a probe that bind your target, and then a transduction that will tell us that the complex is formed. So that also we can do in the core. I put the antibody in red because it's so much application that we can do with antibody. You can have a single chain. I know it's not easy, but we've done it. We can have epitope and use those epitope with the phage to produce more of those antibody. And in cancer lately, we can have those immune checkpoint inhibitor, very important. Also, I will talk about some of the nanobody. Nanobody are some kind of single chain fragment antibody produced in some type of animal. Same thing, if you want to have some new binding affinity of a specific target, we can serve you. We can also create some new functional peptide that bind some domain of RNA or DNA, and also anything that has to do with drug discovery. So this is not an exhaustive list. It can be more than this. Anytime you have a project, you think of something, we can discuss and see how I can better accommodate you with the phage display system. So how do we do this? Anything before we start is good to know what is your direction, your idea, and make a design. I design and I explain to PI what I will do and how to better serve the PI. I can screen candidate binding the 8-mer or the 12-mer library that we have currently. I can expose a probe on the surface by the fusion with a minor code protein for any of the phages. Meaning if you think this is my probe already, and I would like this to be on the surface of the phage, I can serve you. I can use those probe, expose those probe, and then give you the pure phages. So we can produce then the phage display peptide. We can also produce phage display nanobody. At the same time, like we've constructed those library, we can construct your own library. We can have excuse me, if you want to have your own and personal library, so feel free, we can discuss and construct those for you. So I'm just going to give you like an idea, a heads up of what screening using an M13 and a Qβ library will mean. So the library was designed and constructed in a plasmid vector containing a portion of the phage with the minor structural gene. This plasmid can be co-expressed with an helper to produce a huge library of variant. That means I have all the variant of interest, and those variants will be cloned and then used to produce the phages. So what are we doing when we have the library? Any target can be used. So I really say any. If you have a protein and you are wondering what kind of peptide can bind to this protein, we just have to immobilize the protein target, and we pour the library. We wash to remove the unbound, and we can evaluate the specific one, amplify it, and make many round. So I say any target. I remember using some RNA, getting peptide binding RNA, using some protein, getting peptide binding some protein, using some nanoparticle like gold, like cobalt, and so on. So any target can be used. You just have to immobilize the target, pour the library on the surface, select by washing and amplifying, and you can make many rounds of this selection. The only thing that I have to mention here with M13 is that particular replication system. So M13 is a DNA replication system, so it's easy to have a proof reading. That means in terms of round of evolution, it will be slightly limited, but we can select a perfect candidate with the immobilized target. So we have both library, the 8-mer and the 12-mer. So far, we've served 7 PI with our library, and then 13 targets were hit. We have over 50 peptides that were selected, that were identified. So just to give you an idea, since we start in technically 2021, we have been fortunate to serve up to 7 PI. That means they have a target, and I'll use my library. And most of the time, I propose 10 candidates that turn to be 2 or 3. That means they will have common motif. I don't really want to give some detail here, but if you want me to, you can come, we can discuss. I can give you like more detail. So we have the M13 library. It had been used to serve PI, and then we have select 50 new peptides. And again, when we have those peptides, we can go back and have a reconstitution. If you want more than a peptide on the surface, if you want them to be fused with a linker, we can do that on the side. That means this is the result of my selection. 2 major candidates, I can put a linker between the 2 and see if they are binding with high affinity to your target. So that can be done in the core. So this is just one example where a chlamydia that you all know, one of the key part of chlamydia was used to screen peptide from the library. And they want this peptide with the phage to penetrate the bacteria and to be able to lyse or to block this key infectious agent. So they fuse the peptide select from the library with a penetrating peptide, and they could really achieve a lot with this. So that can be done too. Once we have a candidate, we can fuse and have both on the surface and use to maybe answer your question. So this is the M13 that I'm talking about. I will move to the Qβ library and the selection. If you have any question, maybe I can answer. I think something is in the chat. Can you design display protein target specific structure? Yes, why not? So the thing here, it's up to the triple helix. Anytime you have a fixed sequence, honestly, like the nanobody, I will talk about that later. If you provide your sequence, I will use your sequence, I will display your sequence. If you have something in mind, or even some closed sequence, and I can go back to a story of one of the PI, he has a motif that binds RNA. And he is thinking by increasing some arginine in this motif, he can increase the contact with the RNA. And then I display this to the PI, and he is really happy with that. So if you have an idea of some motif that you think may interact with your helix. I can just come and design with your motif aim and give you the phage. And depending on how you want it pure, if you want to go to cell culture, we have the gradient, cesium chloride gradient, that can go to 95, 98% purity. So we remove spore because it's all done in bacteria. We will remove the spore and get back to the pure phages. So yes, this is M13 that we mentioned. We can have the same thing with the Qβ, which is an RNA, and it's slightly complicated. So this is just one application. Back in the time, foot and mouth disease was really disturbing. It was something in livestock disturbing a lot. And my approach was to see if we can use phage to come back to kind of inhibit this virus. So the first thing was to know the major epitope of this virus. I come with two designs. The key portion, I map the key protein that produce antibody, and come with a small portion that was called epitope. And I told myself, maybe this epitope can be more than this or less than that if one go to many rounds of evolution. So I have the wild type epitope like this in the A, and then I synthesize a library. In the library, which was like 1 million, I have the portion in the middle that represent like nucleotide. I just have a combination to have my artificial library. This library was inserted into the entire sequence of my phage. I have to mention that this phage, it's an RNA phage. We have reversed the entire genome and then cloned into a plasmid. And this genome is under the control of T7 and then SP6 at the end. It's a positive and negative. Whether you use the negative strain, the negative RNA, you have the phage. Use the positive, you have the phage. Meaning we can turn it, the genome, close to the end or the beginning under the control of T7, we still have the phage. So the minor code protein, which is an A1, we can use it to fuse the library that I just showed you, right? And that library can be expressed, can use, the DNA will be used to transform the cell, and we have all those phages. So once we have the phage, again, like back to M13, this is the library. So what do? We have the antibody monoclonal from foot and mouth disease. And those monoclonal antibody are bind to the plate by the FC region. So the FAB, it's up. And we block the plate, obvious. And we pour the library on the well containing the monoclonal antibody. And we will wash it. That means we remove the non-bounding, and we will amplify the difference between M13 and Qβ. M13 need acetic elution. One, you wash away those non-desired candidates. You have to amplify those sitting on the target. In this case, the neat thing with Qβ, you don't need to actually have elution. You can just add your cell directly to the target, and it will amplify, because the minor protein we use to display is different from the protein helping to infect. And we can make round and round of this. We went to 6 rounds, and guess what? We've selected something on the 3rd round similar to the motif that represent the major foot and mouth disease epitope. But the difference is just that we don't need the 3rd amino acid. So canonically, the first 2 amino acids were enough to play the same role with the antibody, even better. So we've confirmed this with the neutralization assay. That means we went to a normal animal, produced the antibody with the phage, and check how this was neutralizing the original virus. So this is one thing that we've been also trying to have out with all the area that has a problem with this virus, especially to count the antibody, protective antibody in animal. So that's why I say we can use it as a biosensor. When we have the major epitope, we can use those epitopes to count how protective some antibody are. And this is just an EM, clear EM. You have the phage without a peptide on the surface. And when the peptide is on the surface, you see antibody really binding those epitopes. Just a proof of concept that we have. I did talk about biosensor. If you have question for M13 or the Qβ, I may answer before I move to the biosensor. Okay. So biosensor, this is really new. This is, I was awarded a grant with NSF. We all know how difficult it was, the 2 years of COVID. We couldn't really tell exactly what is the title of antibody from people infect and vaccinated, infect, vaccinated, and reinfect. We couldn't tell how protective the level of antibody was. So I just proposed this because the key thing was to count, to be sure of the protective antibody. The first part of this work was to map the major epitope of COVID, which we did, we have. And then the second part, can we have the major epitope fused there and have a transductor to be able to count those antibodies? So we present the two protein peptides, on the surface. That means I have my probe, I have my linker, and I have the singular sequence that will tell me the complex is done. And I present those two on the surface of the phage. And I will use this in just ELISA to know how many transductor can bind to the specific antibody or the target. I will count, and later I will have a competition between both, like you see here. So if I have antibody binding to these 4, and then you have those 2 still remaining, then I know what the total control look like, and then what the subtraction will look like. And with this, we can easily control, we can monitor some of the protein. We can all do this in the core. We can have like sensing some specific protein using the phage. We just need to have the appropriate transductor. We have nanoparticles that help. We are thinking of FRED, how energy can be when you are in close contact, you transfer energy, you have signal. We are thinking of all those. So we'll come with some mechanistic way of presenting small molecule where you can use even your cell phone out there to monitor some of the molecule. So like I say, in competitive ELISA, it's easy to have this. You have the normal control, and then you can have increased antibody specific. You can count the one that you know, and you can come with a result of subtraction. So we call this biosensor because it can sense, it can tell you the presence of antibody, and it can also tell you the title of those specific antibody, and which is very, very important. So with this, we have up to 15 peptide probe that were displayed, going from 18 to 60, that means to 60 amino acid. We have 2 major biosensors that were developed. The first one was with foot and mouth disease. We can count antibody of foot and mouth disease specific. We can count antibody of SARS-CoV-1, SARS-CoV-2. So those are the 2 major achievements with this technology. Again, some of those papers are still on the way, but we can do it. I will pause a little bit again, and so I will go to nanobody. Any question? Let me look at the chat. No. Yeah, so this is relatively new. After COVID too, people were like, antibody can cure, antibody can help. Then scientists started looking at different types. And there were some animal, camelidae, that in the bloodstream, you have those unique homodimer heavy chain-like antibody that serve as antibody for those species. And it's very small size, monomer, and robust. So we can tailor this and use the naive library of those animals to produce some nanobody with so many different applications, right? And I use one to prove the concept. I use some nanobody that were used to crystallize lysozyme to really just prove the concept. So it looked like this slightly, the nanobody, it was published, but it was just one. And I presented on the phage. And this is the sequence slightly. It's almost 120 amino acid. Because it's an antibody, I just put the his tag at the end. His tag doesn't disturb the portion that was used to crystallize lysozyme. I put the his tag to track it. And you see that it was fused with the minor code protein of my phage. You can really have the histidine tag at the end, and you see here, and those are the stop codon at the end of the phage. And this was expressed on the phage, could give pure phages displaying the nanobody. And we just use a dot plot to check that the histidine was present. And then the crystal structure, we are working with the cryo-EM core to see if we can go back. It's just a proof of concept. We were interested more in, can we have that size display? Yes. Can we have the antibody, the nanobody binding the lysozyme? Yes, we can really see the binding when we have the phage on the plate, have ELISA with the lysozyme and antibody again lysozyme by ELISA. And the plaques, and for those who are not familiar, we count phage with the plaque assay. We make serial of dilution, and then we can tell where we can count. So this is one example. Here was crude. When we have expression, we just test it. And then when we make a dilution, 1 to 10, and you can see it. So another plaque assay for you to have just an idea, the morphology. This is how every day we count those viruses. So far, we have 4 PI that have been served with whatever sequence they provide with clone and give them back, 4 different nanobody. And a library is under construction. I have a company from Germany called Nanotag Biotech. So what are they going to do? This type of animal or close to, they will pull like a library, the naive library of 10 different species, and then cell. They will copy by PCR so we can clone this directly and make a library of phages for the nanobody. Again, in the coming day, we may have this available for any selection. I'm thinking of everything, any target, even cryo-EM, you want to crystallize some of the protein. We can see if they bind to that, what is the affinity, and then can we really use this? So just to cover the 4th part. I didn't want to go to a lot of specific, but this is what we're doing. This is what we can do with the 2 types of phages, not just the selection from the library. We can construct your own library. We can present your own target. We can design anything going to the target. Or if you have the target, you want to map some of the amino acid, we can go to that level. We can have the different mutation of the motif to tell you what are the best or the key amino acid involved in your interaction. We know it's a key. to really be sure that a target is druggable, we should be able to tell what are the major interactions, what are the key amino acids involved in probe and target. So that we can do. Because, like I said at the beginning, the advantage of the phage, you have the genome, the genotype, and then you have the peptide on the surface, the phenotype. This link between the genotype and phenotype is manipulation. We can just change this and see if the binding is still the same. Again, it's not that easy, but I'm just saying it's very possible. We just have a turnover of a month to present something, to have different mutation, and then you can use it to study what you are studying. And I can use one example since we still have some time. The major, I'm going back to what we just have, being an investigator of COVID, the major motif binding the receptor, which is the ACE2, is just 17 amino acid link. So we are trying to know what is really the key one. So if you split them into four amino acid that come this contact surface with the receptor, we really want to go to the question and answer, what is the key amino acid? Not just that, with the time, how is the pocket, how is the binding, the contact between the ACE2 of the rabbit, of the rat, of all those animals that we claim to have the zoonosis from? How is it different from the human one? What are the amino acids in those playing the same role? So just to give you like an idea, this can be done to any intracellular contact between protein and protein. We can narrow down to the key one, or we can present the wild type and you study your binding affinity, and then later we can come and see what are the major amino acids. So please feel free if you have something in mind. And like I say, not just the list that I've provided, not just the first list that I provide, there can be more than that. But I know it's important sometimes to say, this is what I know to be some of the key part of investigation. Mimicking extracellular protein interaction, intracellular protein interaction, enzyme inhibitor, biosensing, antibody, epitope, even the new affinity, right? We are planning with some of our colleagues to see if we can also insert non-amino acid to some of those motifs. They're very important. Yeah, and something I did not mention, there are some cysteine playing a key role in nanoparticle binding. Yeah. So I have a question here. Is it possible to design phage display protein that can interact with this RNA? Yeah, I have with one PI here. He has his RNA, and then he biotinylated. And I use my library to see what are the peptide that can bind this RNA. Again, I can give you candidate, but to really tell which one is better, it's up to you. It may come to 2 or 3, and that's it. So we still have time. We can talk some more.
Alan, maybe you could talk about how do you charge for the service? Is this something that you would prefer? That
oh yeah, a single probe. Yeah, that's true. A single probe. The hour is like 71 and less than the one of Kentucky, and then we can put a probe like 4 or 5 hour service. And the library, initially I was charging for the design, but now I think I can just design, explain to PI, and we're talking about 600 to clone it in. So depending on the time, we can put the hourly, it's a $71. Yeah, I can tell more to who really want to know. It's important with all those rubric, the nano The library is different. The time we spend is different. The probe is different. The nanobody will be different from a single probe. And then the selection. So initially, we have the selection, the sequence. If you want the sequence, you want the reconstitution, the price will be different. If I'm charging you $600 to have a screening, if you want me to put it later on the surface, I may charge you $200 more to have it in different phage. And what I did not mention too, we combine those two systems. So you can have a library here and decide to have it display in RNA, have more evolution, then we clone it back there. So you screen here, you go here, or vice versa. We have both available. Yeah, so feel free. And we combine service. Sometimes people sequence with mass spectral, and then they decide to see if they can synthesize. Lithium will help to synthesize those small peptides as control. If you decide to come with phage, I will just use phage. So that's why we are like really one, right? If you have higher throughput screening and have some target you want to investigate more, yeah, we can combine. You can have your mass spectral, you can have synthesis, you can have phage display. Yeah, that's why we are in one, honestly. Yeah. Okay.
Okay, well, if there's no further questions, thank you, Dr. Wafo, for an excellent presentation. And thank you everyone for coming, and hopefully we'll see you in 2 weeks.